couple of quick announcements before we get started. The first one is here behind me. John and Marcia Salmon-Schulmerich have endowed a scholarship for biology students. If you're interested in working in the medical field after you're done here, here's a lot more information on this. So $2,500, which is not a huge amount of money, but still definitely helpful. Health, career goals, etc. It's a pretty straightforward process. I think the deadline is 1st of February. So if you're at all interested in this, go to the biology website, check it out. Second announcement is appropriate since we're talking about Linus Pauling in the last couple of lectures. Portland's arguably most famous son. The ISEPP, so Institute for Science, Engineering, and Public Policy, has this great seminar series. I don't know how many, have you been to these? Heard about them? No clue, could care less. Seminar speaker coming up this Thursday. I'll be on a plane, so I won't be able to make it. Tech Normality, Survival Guide for Advanced Species is one, but I think most importantly is this guy right here, Eugene Koonin, who's at the NCBI, National Center for Biotechnology Information, basically GenBank, where all the sequences are held. He has some really fascinating theories about viruses. Of course, that's probably why I'm interested in it. He's also a wonderful guy, hardcore marathon runner, and he'll be giving the seminar on the 18th. I think, I'm not absolutely certain, but I'll check on this as of next time, that we can either get very highly discounted or even free tickets as a PSU student, because PSU also sponsors a seminar series. So I'll be going to definitely his talk on Thursday evening. There'll also be a Thursday noon seminar that Eugene will be giving in the biology department here at PSU. We're still trying to figure out what room that's going to be. We're trying to get to a different room than the normal one that we have. But I will keep you posted on when Eugene will actually be here. Last announcement for this morning is that I'm not going to be here on Friday. Dr. Bartlett will be giving a lecture. He will have clicker questions. He'll be forwarding them to me. Hopefully he will post his lecture and give it to me so I can put it on D2L beforehand. I do not think that he's going to be recording it. So you might want to, those of you who do recorders might want to bring them. I did change the frequency today, so hopefully we won't have anyone coming in and giving us a hard time. We shall see. Or maybe I'm just in the dean's office right now as we're speaking. So, well, it's interesting. Maybe we have killed some of the other things. Formatting here is always an interesting issue. So, speaking of clicker questions, there's an interesting change that happened here. But we can still do this. It'll work. Trust me. I think, anyway. So DNA was originally identified as the genetic material by analysis of salmon sperm, determining the relative amounts of C's, A's, T's, and G's, purification of the transforming material, that's D. This is C, D. E, viruses of bacteria. B has just disappeared. So hopefully it's not the right answer. It's still AA. It's a frequency just for my wireless mic that I changed. So I'm not showing up in history classes upstairs, at least hopefully not, as far as they're concerned. So, originally identified as the genetic material. Genetic material, genetic material, genetic material. That's the part that's missing here. DNA was originally identified as the genetic material by X. 5. we have lots of differences of opinion here. So, fortunately, only one person liked B. I'm not sure quite why they like B. Maybe they had wishful thinking. Analysis of salmon sperm, that was actually the second time that they found DNA, but they had no idea whether it was a genetic material or not at that time. That was Friedrich Miechner's work. And at that point, there actually were salmon in Basel, Switzerland, but that's a totally different story. Determining the relative amounts of C's, A's, T's, and G's, you can determine relative amounts of C's, A's, T's, and G's. The first time that people did that, what did they find? They find they're about equal. If they're about equal, is that going to tell you much that it's a genetic material? If anything, it says it's unlikely to be the genetic material. The transforming material was what? DNA, yes. But what was it transforming? Changing bacteria from non-pathogenic to pathogenic, and also from rough to smooth, but that just had to do with the presence of the capsule protein. So it's a changing in terms of pathogenicity. Viruses of bacteria, this was the Chase and Hershey experiment. I much prefer to call it Chase and Hershey because Martha Chase did most of the work. And that was kind of the nail in the coffin. But the really first good evidence for DNA being the genetic material was in the purification of the transforming material. And hopefully the formatting for the next clicker question is going to be a little bit better. That does remind me, most of you have registered your clickers on D2L. That's where they need to be registered. Sorry about that. That's because the company keeps changing things on us. If you go to D2L and see that you don't have a number, 0 through 6, next to your name under clickers, let me know. Send me your Odin username and your clicker ID, and I will link those two together. Just go ahead and send those to me via email. Okay, our next clicker question is fortunately better formatted, and that is, what details of the DNA structure model could Watson and Crick not get from stealing Rosalind Franklin's data? That phosphates were on the outside of the helix, that DNA was helical, that there were 2 repeats in the structure, the DNA strands were antiparallel, or Stedman's favorite, multiple. Ten, 5, more and more clickers each time that we're here. So from Rosalind Franklin's data, they were able to see what? That there was clearly a helical structure to it. There were clearly 2 repeats in the structure. Were they able to see where the phosphates were? No. You can't tell from an X-ray diffraction pattern what atom it is. It'll diffract, but it doesn't tell you what that atom is. Did the DNA strands were antiparallel? Definitely not. So what do we end up with? E, exactly. Which most of us like, which is good. So, moving on. Wanted to briefly talk about some techniques, and this is really critical, partly having to do with the structure of DNA. Unfortunately, they don't do, I think, a nice job of it in our textbook, so I'll try and go relatively slowly. There's plenty of information online about this, but it has to do with again, Watson and Crick's model for complementary strands, anti-parallel to each other, forming up the opposite strand with base pairs between the two. One of the things that we didn't mention last time is that the obvious prediction that you have here is that because you have these two strands, you also always have two copies of your information. And so if you have two copies of the information, it's really conceptually easy to replicate it because you just pull the two strands apart and replicate each one of them. We'll talk more about that next week, exactly how that happens. But in order for that to be able to happen, you've got to be able to pull strands apart and put them back together. And so the critical thing in terms of pretty much all molecular biology as we know it, all the human genome sequences, you name it, has to do with pulling DNA strands apart and putting DNA strands back together. And so that gets us to thinking about how can you tell that DNA strands have come apart and gone back together. The way that this happens is basically if you just look at the absorbance, put your DNA in a test tube or usually a cuvette, shine UV light through it at 260 nanometers. If you have double-stranded DNA at a certain concentration, random concentration, in this case is 50 micrograms per milliliter, you have an absorbance of 1 at 260 nanometers if you have double-stranded DNA. However, you pull the two strands apart, you have a larger absorbance at 260 nanometers. And that has to do with the structure, all the base stacking that happens in the inside of your double-stranded DNA that you don't have as with single-stranded DNA. Why is this so important? Well, it allows us to look at DNA coming apart and going back together. There are lots of ways of doing this. The easy one is with your little cuvette with the DNA in it, just cranking up the temperature. And each time you crank up the temperature, you measure what the absorbance is. So that's all that this is here. So this is your absorbance at 260 nanometers. We have less absorbance of double-stranded DNA, more absorbance of single-stranded DNA, and just heat up the DNA. There are a couple of interesting things about this. One is that DNA is pretty darn stable. You can heat it up to 70 degrees. It's pretty standard E. coli DNA. Up to 70 degrees, nothing happens. But if you heat it up enough, the two strands will come apart, and that's due to breaking of the hydrogen bonds. So that's one aspect. You can heat up your DNA and the things will come apart. More interesting is that this transition between double-stranded and single-stranded is a really rapid transition. It happens over a relatively short range of temperatures. And what that means is once you start to pull the two strands apart, it goes really fast. And the actual temperature at which you have half of this take place is what's called the melting temperature of your DNA, or TM. That is completely dependent on the, as long as you keep everything else the same, the GC content of your DNA. So here, we now just look at the percent GC and the melting temperature. Do have one DNA of one particular percent GC, another DNA of a different percentage GC, measure that transition point between double-stranded and single-stranded, and you can plot really easily either knowing the TM or knowing the percent GC. You can get either melting temperature, which is easy to get, or the percent GC, which is a lot harder to get unless you've done some sequencing. Basically, to help beat on the dead horse here, any kind of double-stranded DNA, as long as it's completely double-stranded, it's going to be very stable until you heat it past a particular point where things start to come apart. And then that's going to go really quickly. And so you can see here by the bottom, anytime you don't have good base pairing, this is a not stable interaction. If you have good base pairing, it's going to be a very stable interaction. One of the other really fascinating things that people found in studying these things is it's not just taking the two strands apart, but those two strands can come back together. And they come back together very quickly and very specifically. And that allows us to basically do all the other experiments we'll be talking about pretty much for the rest of the course, but certainly for a little bit more of the rest of today. So any more questions about sort of DNA structure, DNA, pretty straight biophysics stuff? Yeah?
So you were saying if there's good base pairing, they'll come back together after being denatured quickly?
Okay, so the question here is, am I saying that if you have good base pairing, and I'll paraphrase good in just a second, do you have things coming back together rapidly? And the answer is yes, but good base pairing basically means that you've got almost 100% matches on either side. They're completely complementary to each other. And it turns out you can do exactly that same kind of graph that I showed here with the number of mismatches and get very similar numbers that way. But that mismatch only goes to about 90% or so. If it's more than 90%, you're going to have a really hard time getting those two strands to come back together. There's another question over here, I think. Yeah?
So if you heat it up to break it apart, how do you get it to go back together?
You just cool it down. So the question is, if you heat it up to break it apart, how do you get it to come back together? Just cool it down. So it's really amazing how well DNA does that. It does find its partner really, really quickly. Pardon? So allowing it to, the question is allowing it to cool or physically cooling it. So it does depend a very little bit on the rate, how fast you do it. If you actually put them together really, really fast, and we'll talk about this a little bit more when we talk about genome complexity, sometimes they find the wrong places. But as long as it's relatively slow, they will find the right place. Amazingly, even if you have the whole human genome, for instance, 6 billion base pairs, and we'll see here for some of the oligonucleotides, 20-odd nucleotides, they'll come together, and they'll come together extremely specifically. Yeah?
Just for clarity, so you said the percent of G and C, does that exclude or include A and T?
Okay, so the question here is percent GC. When I say percent GC, what does that mean about AT? Well, the great thing is that you're going to always add up to 1. So if you've got 60% GC, you could also do exactly the same thing and say, so it's 40% AT.
Right, I was just making sure there wasn't an exclusion there.
No, there's not a particular exclusion there.
But the graph would be inverted if it was percent AT, correct?
If you're, well, because of course they can be increasing the GC. Yes, so they'll have a different graph when you look at that. Be a great thing to do to study for this. Plot it out. Bring it next time. Yeah?
Why does the percent GC matter? Is it like stronger bonds than ANT, has stronger hydrogen bonds or something that make it more stable?
Yeah, so the question is, why does percent GC make a difference? It seems to be mostly the fact that with GC you've got 3 hydrogen bonds, and AT you only have 2. That's slight oversimplification, but we don't want to get into too many details. If you're interested in that, take Biochemistry 492, I think, where they talk about nucleic acids. Really cool course. Dr. Lehman, chemistry, does a great job with it. A little beyond the scope of this course. Also a bit why you get different absorbances with single-strand and double-strand as well. Okay, so that's standard DNA. Of course, as I mentioned last time, Watson and Crick were wrong. That was one of the other clicker questions that I had. Because if all of our DNA were like this, the length of DNA in each and every single one of my or your cells would be about this long, from the floor to here. That's going to be a problem. So somehow you have to compact that DNA. DNA, because it's only got those 4 bases, and because you've always got 2 copies of it, is ridiculously inefficient in terms of encoding genetic information. So you have to have ways of compacting it. The way that works, of course, is through chromosomes. Chromosomes, bacteria have chromosomes as well. A lot of people forget that. Archaea have chromosomes as well. Normally they just have 1. Of course, in humans we have considerably more. So look at karyotypes a little bit, particularly human chromosomes. Depending on how far we get today, we'll talk about the human genome. Maybe Dr. Bartlett will talk about it more on Friday. But most important, for the next half hour or so, we're going to talk about how to do that compaction, how to go from almost 2 meters in length down to a few microns, which is what you have inside nuclei. main thing, of course, that does that are the nucleosomes. So we'll talk about some of the histones. This is really well understood, how that first step of compaction happens. Beyond that, it starts to get a lot more speculative. And so we'll talk a little bit more about some of that speculation. Chromatin is just another name for the DNA plus the proteins that come together. There are plenty of proteins in eukaryotic chromatin that are not histones. So there are lots of these, what people call non-histone chromatin proteins. They're just what's present bound to the DNA. So chromatin is a way of packaging your DNA and compacting it. So obviously it's the first thing that has to happen, is you have to have this big compaction that takes place. The other problem is that, as we talked about last time, we talked about the major groove, you have to be able to access the information somehow that's in the DNA molecule. Well, how can you do that if it's ridiculously compacted? So that compaction process can't just be completely squish everything down. You have to be able to access some parts of that as well. And so that's the really, I think, almost most interesting aspect about chromosome condensation, is having this great compaction, but at the same time having the availability for interaction with the DNA. If your genetic material is just completely crushed, you can't do anything with it. You can't even replicate it. So it's that balance between compaction and access, which is a really interesting question that we still don't completely understand exactly how that's working. As well, you get some protection of the DNA by this interaction with proteins. We talk about DNA repair again later on in the course. We'll see, as I always say, DNA is a really crummy genetic material, A, because it's inefficient, as we just talked about, but also because it's susceptible to all kinds of damage, which will change the genetic information. We'll talk about how that happens. Organization. Hopefully everybody knows about cell division, mitosis, chromosome compaction, pulling those chromosomes apart. That's a way of organizing your DNA and also allowing that separation to take place. It's not just the DNA that does that, it's the proteins that are associated with the DNA. But first, let's talk about compaction. Again, as we started out with, here's our nice model form of DNA. Again, lots of mistakes. Some people came up and found that the other day. That can't be what's inside the cell. In the most compacted state, which is about 10,000 times, that's in your metaphase chromosomes. This is the absolute complete compaction. As it turns out, metaphase chromosomes are not active. They have been compressed so much that you don't have that access anymore. On the other hand, most of the time, and most of the cell cycle, you've compacted about 1,000 times. And that gives you just about enough to get inside a nucleus. Varies from different parts of your genome to the other. But for the most part, most of the DNA in most cells is going to be in this really poorly defined 30 nanometer fiber. So just 30 nanometers across. How far across is this molecule? 2. So, 15 times of these. So it'd be about that wide, relatively speaking. That's what most of the DNA in the cell, and that turns out is a pretty active form of your chromatin. When I say active, that means you can have transcription, you can have interaction with other proteins, you can have replication. That's what's happening with your 30 nanometer filaments. You can get more release of the DNA from the proteins, and that gives you these so-called 10 nanometer filaments, and each of these little black blobs here, or gray blobs, represents a nucleosome. And we'll talk about those nucleosomes a little bit more later on as we move through. But first I want to talk a little bit about these metaphase chromosomes, because this is how we can in fact visualize the genetic material. In fact, these were found way before people knew that DNA was in fact genetic material. They found chromosomes, which were mostly made up of protein, if you purify them. So again, that's why people thought that protein was genetic material to start with. Turns out it's, of course, the DNA plus these proteins. This is a standard, although actually for the minority of us, karyotype, And the karyotype is just the chromosomes that we have. In this case, it's the unique chromosomes. Most of the time, we're going to be diploid. Here is just a single copy. 22 autosomes, which are the non-sex chromosomes, and the 2 sex chromosomes, in this case, XY individual, so a male. This is how you almost always see karyotypes. What they call distinct banding patterns. So you've got dark here, light here, dark there, light here, dark there. What does it mean? Nobody knows. But we do have clear constrictions right in the middle, in the middle, more or less, of any of the chromosomes. And that's where chromosome division and separation is going to take place. That's what's called the centromeres. That's that separation. Of course, it's all proteins which are involved in this process. Some chromosomes have these little knobs at one end, so they're drawn. This looks really nice, but this is not really what karyotypes look like. Oh, sorry. This is this is what a karyotype really looks like. In fact, you get a nucleus that kind of looks like this, and you pull out the various different chromosomes, and they look like this. Like, whoa, okay, distinct banding patterns. How do you go from this to this? You're really good at it, basically. And so people can line all these things up. Another way of doing this is what's called rainbow or painting stains. This gets back to the whole pulling strands apart and putting them back together. So the way that you do this particular kind of experiment is you take all those chromosomes, these are metaphase chromosomes, and heat them up together with lots of little pieces of DNA with different fluorescent tags on them. And those little pieces of DNA have sequences which are complementary to different sequences on any one of these different chromosomes. Then you cool them down slowly, or relatively slowly. In this case, it doesn't have to be that slowly. And then look at them under a fluorescence microscope. And you can see that some of them are yellow, some of them are light green, some are dark green. And this also allows you to relatively quickly identify that this is one of chromosome 1, this is another one of chromosome 1. And you can walk your way all the way through here and end up with your 22 autosomes together with the sex chromosomes. This is a normal karyotype. Why do people care so much about karyotypes? Well, it turns out in a number of cases, you have DNA which is moving around all over the place. In this particular case is what's called a translocation. What happens is you have a piece of one chromosome that's gotten moved over to another chromosome. Why do we care? Genetic defects is one thing. Turns out that this particular translocation, which is shown here, generates what's called the Philadelphia chromosome, which leads to development of chronic myelogenous leukemia, which is the disease for which Brian Drucker, here at OHSU, developed Gleevec, the drug which blocks, actually, the protein which is misregulated due to this chromosome translocation. So just by finding someone who has this chromosome translocation, we now have a drug that can treat that. It's pretty darn amazing, as far as I'm concerned. So the other thing, as far as karyotypes are concerned, is this is a normal karyotype. Male or female here? Female, because we've got 2 X's. Again, how you can line these up, it still blows me away how people can figure this out. What happens in lots of cancers, above and beyond the CML, which is pretty straightforward, is you end up with really funky karyotypes. So this is another karyotype, again, courtesy of the OHSU cytogenetics lab. This particular cell has 5 copies of chromosome 1, 1 of 13, none of 17, So this is a really classic kind of problem that happens in cancer cells. Now we're getting much better at sequencing the whole genome of a lot of these cancer cells. But this is kind of an indication of some of the things that go wrong in cancerous development. Lots of chromosome problems. Yeah?
Those 5 chromosomes between 3 and 4, are those just so messed up they didn't fit anywhere, or are they trying to be part of 3?
That's a good question. I'm not sure if they were able to identify these or whether they're 3 or not. I'm not good enough to read the bands on there to figure out what it is. Yeah?
So when you sequence a genome,
Yeah.
does it just start at the start of number 1 and then go to number 2, 3, and 4 just in a straight line?
Okay, so the question is, how do you sequence a genome, basically? Or how do people sequence the human genome? We may get to that today. I don't want to get into too much detail now. If not, we'll definitely get to it at the end of class when we talk about DNA sequencing a little bit later on. The basic answer is you break it up into literally millions of little pieces, sequence all those little pieces, and then put them all back together. Yeah?
So going back into the translocation that results
Yeah.
from the leukemia, translocation results from the chromosomes becoming entangled. So how do you prevent that?
Okay, so the question basically is how do you prevent this translocation happening? And because it turns out it's due to crossing over that we'll talk about when we talk about recombination a little bit later on. Turns out that some of that recombination, at least between sister chromatomes, which you have the 2 copies which are more or less the same, moms and dads, that is actually required. This is just a mistranslocation that happens in some cases. Turns out we don't know any way to control that. There's no particular way. It just happens, fortunately, relatively infrequently. And there are also things that we're not going to get into this class, error checking mechanisms to try and make sure that if that does happen, that particular cell dies. And what the problem is when you have these cancer cells, those error checking mechanisms have been blocked or are no longer functional. Yeah, so this is not only really strange numbers of chromosomes in lots of places, but also a lot of translocations are happening here as well. Here's another cancer karyotype. Even more bizarre, this one's completely missing number 14. Lots of cases you just got 1 copy of the chromosome. Again, how you can identify the banding patterns on here, these guys are way better at it than I am. So let's look at some normal chromosomes. This is an example of what the genome looks like after we finish sequencing it. And again, we'll talk about that more later. Turns out this is the first whole chromosome of the human genome to be sequenced, chromosome 22, relatively small, only about 50 million base pairs in length. If you look at about 10% of one of that, one of the copies here is that 2 sister chromatids right before meiosis. About 10% of that's got about 40 genes in it. 1% of that, of course, got 4. And then if you keep zooming in to each of those individual genes, one of the surprises when they did the human genome is it turns out there's a lot of junk or at least non-protein coding DNA in the human genome. That's basically shown down here at the bottom. All of these red bits, which if I can get my mouse to come over here would be nice. These red bits are the bits which are actually encoding protein. Everything else, all the gray, is not encoding protein. And the yellow here are all what people call regulatory DNA. That's where we have binding of regulatory proteins which control whether this gene is turned on or turned off. And that we'll talk about much more towards the end of the course. But here, that's that one protein and gives you eventually this final folded protein, which of course has the activity down here. There are about 20,000, we'll take a look at this in the next slide, of these protein coding genes in the human genome, which is a pretty small number. But that's because there's so much which is non-coding. So either these bits in one particular gene, and these are all the introns, the exons are what's coding for the protein. These exons can actually vary anywhere from 8, and the average is 145 base pairs. So they're really very short. These introns can be thousands of base pairs themselves. And so that process, we'll talk about splicing and how you go from this, people call primary sequence and primary transcript, to the actual protein much later on. The other thing again here, hopefully this is kind of beating on the dead horse, these genes here also are separated by big regions of DNA that, as far as we can tell, don't code for anything, certainly don't code for protein, but there are very large numbers of them. And again, this is a relatively small chromosome at 50 million base pairs in length. If one were to take one base pair and make it 1 millimeter, the length of the human genome will be all the way across Africa. So again, the sizes here are really pretty amazing. Vital statistics for the human genome. You've probably heard me talk about 3 or 6 billion base pairs for the human genome. That is a number I would expect you to remember. Pretty straightforward. There are not too many of them. I would not, however, try and remember the number of genes coding for proteins, because it keeps going down. The original estimates that people made for the human genome, telling you how old I was, or am, I should say, still relatively old when that happened too. First, people thought there would be 100,000 genes in the human genome, protein-coding genes. And in fact, people were selling the sequences for these 100,000 genes in the human genome. And then they sequenced them, and they found out, actually, no, it's probably on the order of 20,000 protein-coding genes. Yeah? The DNA length, yes. So approximately 3 billion base pairs. That's a single copy. So that's the haploid copy. Diploid is going to be twice that. So 2 times 3 is 6. That's about the level of math I expect you to know for this class. So mentioned some of these things before. The other numbers here are really not critical. But just as a bit of an idea, some genes are millions of base pairs in length. Usually they're on about the size of 30,000 nucleotides. Some have 1 exon. Some have a whole bunch of exons. Exons, they say, vary hugely between, I should say introns, I should say, vary hugely. Exons do, but a lot less, about 145. We'll come back to this 145 number. So numbers I say a whole bunch, like 145, 145, 145, 145, might be good numbers to remember. Turns out that that's almost exactly the number of nucleotides that you need to encode a domain. What does a domain do? Has a particular structure, which is going to have a particular function. So very often, these 1 exons are going to encode 1 domain. And we'll get back to that later on in the course. The number which is totally wrong on here, even though this is a 2015 publication, approximately 9,000 non-coding RNA genes. I think we're above 20,000 now. Just to give you a bit of an idea, where we're at here. Certainly more than 20,000 pseudogenes. Another number that I'm going to use a lot, so it might be a good one to remember, is percentage of DNA sequences that are protein coding in the whole human genome, 1.5%. Very small number. So how much is then not coding for proteins? 98.5%. At least not for these unique proteins. Yes?
So when it says there's such a small difference between us and our relative ancestor, like the primate, right? And only 1.5%, right? What's the difference between us and primate?
So the question is, what makes us different from, say, chimpanzees, which genetically are our most closely related relative? And you say 99%, 99.5%. Unfortunately, the devil's really in the details about exactly how you do that. And it's also a very open area of research to try and figure out what's going on there. If you look at the protein coding sequences, that 1.5%, that's where we're 99+% identical with these. Yeah. So it's in some of the non-coding sequences, there's less identity. But whether that makes the difference between us and chimpanzees or not, who's going to do the experiment of taking them and switching them? So the question is this 1.5% protein coding stable or changing a lot? Actually, it's a very stable number. That's been 1.5% basically since we got the coding sequence back. So that has really not changed at all. The other last thing here is lots and lots of repeated DNA elements in the genome. So about 50% of our genome is repeated sequences. I will argue that, of course, it's all viruses, but that's a different story that we can talk about later. Okay, so let's move on, talk about the nucleosomes and chromatin. Talked about this a little bit already. It's just exactly the same figure we had before. 30 nanometer filament. Again, most of your DNA in an interphase nucleus is going to be in this form. Some of it, and the most exposed, is going to be in this form here. So each of these little blobs that are connected to each other by the DNA. What are those blobs? They're nucleosomes. Nucleosome is made up of approximately 150, actually varies a little bit from species to species, base pairs of DNA wrapped around 8 proteins. And what I wanted you to get out of this particular slide is that this is a very symmetric structure. So if you look at one side of the nucleosome, you flip it over 180 degrees, it looks extremely similar. The ends of the DNA are very close to each other. They're right at the top here in this view, right here. And what that means is that this is just really a way of compacting your DNA. Another interesting thing about nucleosomes is that you'll notice that compaction is taking place through proteins that are on the inside. The DNA is on the outside. This is great from what we talked about before. You're getting compaction, but you're also having access. Because there's access now to the DNA that's present on the outside of these individual nucleosomes. How do you get nucleosomes that form? It's due to the presence of 4 histones. They're almost always coded here in blue and green. These are Histone H3 and Histone H4, and H2A and H2B, which are going to be yellow and red. So H3 and H4, you've got 2 copies of each of those. H2A and H2B, you've got 2 copies of each of those. In the cartoons, as we go through, they try and do this for the whole textbook, have those same color scheme as we move through. And so when you see that, that's where that's going to be coming from. How do you have interactions? How do you get the DNA wrapped around these? It's basically lots and lots of different interactions, lots and lots of weak interactions, van der Waals interactions, hydrogen bonding interactions, electrostatic interactions. But one of the things that came out of the X-ray crystal structure of the nucleosome, which was the largest structure to have been determined at a molecular resolution at the time, late 1990s, one of the things that they found was that almost all of the interactions between the histones and the DNA in the nucleosome was, not surprisingly, to the backbone. Why it's not surprising that those interactions are with the backbone? Because you don't want to have specific interactions. If you had specific interactions of your histones with different DNAs all over the place, you'd have to have literally millions of different histones, which are going to be interacting sequenced specifically with the DNA. If you have histones that can interact in a non-specific way with the DNA, then you can use one set of histones for everything, or almost everything. And so that's what was seen in the crystal structure. Here are just a couple of the positively charged side chains coming off of H3 and H4, interacting with the phosphates on the backbone of DNA. How does this actually work when you put everything together? As I mentioned before, we've got the blue and the green, Histone H3 and Histone H4. The main part of these histones are alpha helices. There are 3 alpha helices that interact with each other through an almost coiled-coil interaction. You see these 2 alpha helices here, long ones in the middle, they wrap around each other. So you have H3 and H4. These guys make a dimer. And we talked about dimers before. It's that quaternary interaction. Those come together to make a tetramer. That then wraps the DNA around it, and then you add H2A and H2B and end up with this structure. Again, this is mostly non-specific interactions that happen here. Looks like a pretty complicated process to put it all together, right? Turns out it is a rather complicated process to put it together. So there are a number of extra proteins that help in this process. And so we'll talk a lot about helper proteins, the chaperones, which hopefully you've heard about before from protein folding, how you get assembly of these macromolecular structures. Almost always you're going to have extra proteins that are involved in making sure that you get the right assembly of your structure. As well, as you hopefully have noticed, as this, what's also called the histone core, so it's those 3 alpha helices that interact with each other, there are tails. Again, molecular biologists don't have much of a sense of humor when they put together these things. The geneticists are way better. They've got great names for things. We'll get to those later. But you have the tails, which are not part of the histone fold. So if you'll see here for histone H4, you've got this long piece on the N-terminus. Histone H3, you've got a long piece on the C-terminus. When you assemble the nucleosome, these guys hang out on the outside. They reach out from this nucleosome core structure. And as Dr. Bartlett will talk about later, very important for modifying these histones, modifying the histone DNA interactions. So again, same kind of thing here. We've got 2 H3 and H4s, 2 H2As and H2Bs. They all come together like this. If you treat with a small amount of protease that will cleave off anything which is not protected by DNA, you can cut off these tails relative to each other. So now I want to talk about some of the experiments that people have done to show nucleosomes, how they actually are assembled together. The way that this was done originally was by purifying that 10 nanometer filament. You remember the black blobs on sort of the gray line. Treating with very small amounts of a nuclease. Again, ACE means what? Enzyme, usually chopping something up. So in this case, nuclease. It's an enzyme which is breaking up your nucleotides. In this case, it chews up all of the DNA that's not protected by proteins. Then you have these individual nucleosome particles. If you now remove the protein from the DNA, you can now see that you've got about 145, again this varies from species to species, nucleotides that was protected by the histones. You can also now take these histones, break them apart from each other, and that's how we know that we've got 2 H2As, 2 H2Bs, 2 H3s, and 2 H4s. Talked about the tails and the histone core as well. Here's that histone core, 3 alpha helices. This one's going to wrap around that one to give you the histone core structure. All of these guys have tails. H2A has a tail, H2B has a tail, H3 and H4. We're going to talk mostly about H3 and H4 in terms of these modifications. So these guys get assembled into nucleosomes. Nucleosomes are nice because the DNA is present on the outside and you have some compaction, but clearly when the DNA is wrapped around, you're going to have some parts of that DNA that you're still going to have to access at some point. So how does that happen? There are also, not surprisingly, a number of proteins that help with that. And probably the best known of those are what are called the chromatin remodeling complexes. Whenever you see complex, that probably means multiple different proteins that have come together. That's also clearly the case for these chromatin remodeling complexes. We'll talk much more about these as we move on through the course because it turns out they're very important for regulation of what DNA is available, what DNA is not available. All these chromatin remodeling complexes depend on ATP hydrolysis, and basically all that they do is use ATP hydrolysis to move the DNA relative to the histones that are present in the core. And so what is trying to be shown here is here's your DNA, here's one particular sequence on your DNA, here's another sequence, and here's another sequence. The interaction of this chromatin remodeling complex and ATP hydrolysis basically turns around the nucleosome. So this DNA sequence comes out, this DNA sequence comes in a little bit. These are big complexes. This is, in fact, the structure that was solved about 2 or 3 years ago now of one of these histone remodeling complexes together with a histone. So they're really big proteins in terms of the nucleosomes. Here's another one of those different histone remodeling complex which is there. I mentioned that you have usually almost all nonspecific binding that's interacting between your histones and the DNA. That's not always completely true. Turns out that you can get some DNA sequences that are going to bind to histones a lot better than other DNA sequences. And the reason for that is we're wrapping the DNA around this complex of histones in the middle. Turns out some DNA is easier to wrap than other DNA. And that easier to wrap turns out to be when you have AT base pairs. They're a little bit easier to squish because you only have 2 hydrogen bonds instead of 3. So that squishability allows those to be compressed a little bit. So if you have these ATs or TAs spread one turn of the DNA helix away from each other, which means how many nucleotides? How many nucleotides in one turn? The helix 10, so about 10 nucleotides apart, you can get better binding of nucleosomes to those particular sequences. And you can, in fact, design DNA that's got AT base pairs every 10 nucleotides, which will bind to nucleosomes and bind actually really well to nucleosomes. So if you want to study nucleosome DNA interactions, you'll make a piece of DNA that every 10 nucleotides will have AT base pairs. It turns out that's exactly what they did to get the crystal structure of the nucleosomes in the first place. But even so, you'll also notice that here, that's our minor groove because that's where you're getting the compression that's taking place. But you also have the major groove right here. Well, if you have a major groove here that you want to have interacting with other proteins, because we said that's where all the information is, somehow this major groove needs to be made available. How does it happen? You can have chromatin remodeling complexes, but we also talked about those. Those always need ATP in order to get the nucleosomes to move along. What people have found, somewhat to their surprise, is that the nucleosome structure in and of itself is not very stable, and there's what's called nucleosome breathing. And that is that even though you have, most of the time, 250 milliseconds here, talking about a second, most of the time it's wrapped, but for some small percentage of the time, 5 up to maybe 20% of the time, there'll be a little bit of unwrapping, and then it rewraps. Unwrap, rewrap, unwrap, rewrap. So this is what's called nucleosome breathing. And so the DNA comes off, goes on, comes off, goes on, comes off, goes on. If you happen to have a protein around that can bind to this usually major groove of DNA, which is otherwise compacted inside the nucleosome, just because of this breathing, you can have some interactions that take place there. And we already talked about chromatin remodeling complexes. The last thing that I wanted to talk about in terms of just the nucleosome process is, as well as these chromatin remodeling complexes, you also have exchange of some of the histones in nucleosome complexes. The most obvious time that you need this is during replication. So you have a DNA that's got nucleosomes associated with it. You replicate that DNA. Now you've got 2 copies, but only 1 copy of your nucleosome, your proteins. So how do you make sure that that new copy also gets bound? Because you've got to compact it. It's got to be accessible as well. So the process of putting on those new nucleosomes depends on these histone chaperones. So they'll grab an H2A, H2B dimer and put it into a structure, in this particular case, or in some cases, they'll actually dissociate the nucleosome completely, and we have a complete exchange of nucleosomes. Turns out that this is really important for that centromere. Remember the little piece in the middle, as it were, of the metaphase chromosomes? That's where you have the division of chromosomes that takes place in mitosis. Turns out you have special nucleosomes, which bind to that particular part of your chromosome, which then will hook up to all the spindles, which will allow cell division to take place. So the vast majority of nucleosomes are going to be H3, H4, H2A, and H2B, but you can also have alternative nucleosomes that have to be put in through these chaperones, almost all of which are also ATP dependent. So ATP hydrolysis, what do you think that's doing? What's ATP hydrolysis doing? Giving us some kind of function, right? So it could be making a unstable intermediate, if we're talking about making bonds. But are we making bonds here? Making covalent bonds? No. So what do you think that ATP hydrolysis is doing? It's causing some other kind of change. What kind of change do you get when you have GTP hydrolysis in a G protein? Change in structure. So what's happening on ATP hydrolysis? You have a change in structures. You have a change in structure of your chromatin remodeling complex. You have a change in structure of your histone chaperone. And that change in structure will help then with assembly, disassembly of your various histones. So, as I mentioned before, again, don't have very much of this 10 nanometer filament. Most of the time, we're in a 30 nanometer filament. How do you go from just the individual nucleosomes to what we have most of the time, 30 nanometer filament? The first thing to do is add another histone, Histone H1. So we talked about H2A, H2B, H3, and H4. You know, somebody skipped one, right? Histone H1. Turns out the Histone H1 is also present at very high concentrations in chromatin. But curiously enough, Histone H1 is not like all the other ones, H2A, H2B, H3, and H4. It doesn't have that alpha helical structure with the tails on the ends. Turns out that H1s vary a huge amount between different species, whereas H3, H4, H2A, and H2B basically have almost no changes in them whatsoever. The amino acids that you find in histones, at least the core histones, are basically identical all the way across all of evolution as we know it. So that means it's probably really important for their function. So H1 sits between individual nucleosomes and helps hold them together. We already talked about tails that are hanging out. Turns out the tails of individual histones can interact with nucleosomes right next to them, and that forms this 30 nanometer filament. Everything we're going to look at now are just models. We don't have a very good handle on exactly how these things are functioning. Here we have two nucleosomes with this wrapping around them. This is a model for people, how people think they work. Here's another different model. The last one here, and this is the one that I like the best, is with basically 6 nucleosomes all wrapped together. That makes sense if you have your 2 nanometers here, 6 nucleosomes. 6 times 2 is 12. We're talking about 30 nanometer filaments. So now we have wrapped all around with a bit of a space in the middle. This is a crystal structure of these nucleosomes all come together. This may or may not be exactly what's going on inside the cell. Histone H1 binds to the nucleosome on the outside, helps pull these nucleosomes together, probably also critical for the 30 nanometer filament, and these tails are probably interacting across between the histones themselves. Dr. Bartlett was going to talk about how you go from 30 nanometer filaments to even larger forms of DNA. These are larger domains in scaffolding. So again, he'll talk about those. I want to spend the last 5 minutes on the question that you mentioned about the human genome. And again, these are my wonderful pictures which are disappearing. These are the 2 covers of Nature and Science in February 2001, which is when the human genome sequence was done. Well, actually, it wasn't really quite done. It's when the 2 competing groups that were working, there was a commercial company and the International Human Genome Consortium, were competing with each other in order to try and finish the human genome first. again, basically breaking it up and putting it back together. What did that genome come from? Volunteers of different racial backgrounds were used to make the human genome. Anybody know what most of the commercial genome was? Was it the CEO of the company? Most of the genome that they then sequenced. The latest version of the genome, and no, you do not need to remember this number, but 3,547,121,844 base pairs as of October of last year. Still 271 gaps, so places where we don't know what the sequences are. We can talk about those guys a little bit later on. But again, we've got two groups that did this. It was the good guys, as far as I'm concerned. The Human Genome Project, what they did, and this is getting back to your question about how the genomes were sequenced, is basically they split up the genome into relatively big pieces, about 1 million base pairs each, between 1 million and about 5 million, and then took all of those million base pair pieces, sequenced each of those, and then put them all back together by overlapping each of those million base pair sequences. And the way they did that is through something called bacterial artificial chromosomes. So they basically made a bacteria that had a whole bunch of sequence of the human genome in it, purified the DNA from that bacteria, then sequenced that million bases, and then one group did 1 million, the next group did another million, and so on and so forth, and then they put them all back together. The bad guys, again, the company that was doing this, full disclosure, I started a company about last year, so all companies are not that bad. Although I don't have anywhere near as much money as these guys do. Cellular Genomics basically said, we're not going to do this million base pair piece thing. We're going to do 1,000 base pair pieces and put all those together. If you think about that a little bit, those 1,000 base pair pieces are going to be a lot harder to put back together. And it probably the big reason that Cellular Genomics was even able to do this was because they had, I think, the 3rd fastest supercomputer in the world at the company working on this at that particular time to put it all together. But as it turns out, they ended up using the human genome maps as well, these million base pair pieces that they put theirs back together too. So that needed that. But again, at that point in 2001, there was still about between 10 and 30% that was missing. Now we're down to 99% of coding regions still have about 20% missing sequence. All those missing sequences are around the centromeres and telomeres. So we don't actually know the sequence of a lot of these. Very last slide. What are we at? Number of protein coding genes. Again, you don't need to know these numbers. We're at 20,313. That changes every single time you go back and look at it. So it's not like the 1.5% that doesn't change much. We're now at 25,000 non-coding genes. And these are going to be almost all genes that are going to be coding for RNAs. Be small, long, or miscellaneous. Again, we can talk much more about that. Amazing thing here to me is we've got about 45,000 genes, 200,000 transcripts, so RNAs that are being made. Wait a minute, if you've only got 40,000 genes, why are you making 200,000 transcripts? We don't know, is the answer to that question. We really don't have a good idea. And probably the most amazing thing, getting back to that question about chimpanzees and humans and so on and so forth, if you look at those genes, the 20-odd thousand genes, only about 2,000 of those genes, only about 10% of the genes that are in our human genome are unique to even primates, let alone things like Drosophila or C. elegans.
10% of the 1.5%?
Yeah, so of the actual genes, so they're 1.5% coding, which give you about 20,000 genes. Of those, that's 1.5% the DNA sequence. Yeah. Then of those 20,000 genes, which are protein coding genes, of those, only about 2,000 are unique. So about 10% are unique relative to, at that point, it was non-human, any of the other non-human sequences. We didn't even have non-human primates at the time. The Neanderthal genome sequence was finished about 1.5 years ago. Very, very similar to ours. In fact, so similar in some cases there was probably some genetic transfer between those two. Last thing, yes, very few genes, but some of these can make a whole bunch of different proteins. And so maybe there's a saving grace, and that has to do with how those introns and exons are actually used. Remember little red stripes? Sometimes you use them, sometimes you don't. We'll talk a lot more about that when we talk about splicing later on. Have a great weekend. I'll see you on Wednesday. Monday is no school, Martin Luther King Day.